Hello, this is the first video of the Wet Lab series, which will be published in parallel with Dry Lab series to cover most needed topics for any biotechnologist. In this video, we will talk about an important technique in molecular biology, which is PCR. PCR stands for polymerase chain reaction, and it's used mainly to amplify a specific segment of DNA, meaning that after the first cycle, the DNA molecule is doubled by giving 2. And the next cycle also gives 2, and so on. This technique mainly relies on an enzyme named Taq polymerase, which has the same function of DNA polymerase, but it can tolerate higher temperatures as it's isolated from the thermophilic bacteria Thermus aquaticus, which can withstand up to 96 Celsius. Generally, there are more than 35 PCR types. Each has a different use, but they share similar basic components present in any PCR type. In order to do PCR, the PCR tube must comprise the DNA template, which is the DNA segment we want to amplify, the primers, which are oligonucleotides at which the DNA polymerase initiate its function and build the new strand. Thirdly, we need dNTPs, which are the nucleotides used by DNA polymerase to build the new strand. The fourth thing that needs to be present is the buffer to maintain the optimum pH during the reaction, which is mostly 8 to 9.5. After putting all your components, put your PCR tube in the PCR machine, also named thermocycler. Its main function is changing the temperature in order to complete each step. PCR has 3 main steps, which are denaturation, annealing, and extension. The first step, which is denaturation, is done at very high temperatures, up to 94 Celsius, and it's done to disrupt the hydrogen bonds between bases, thus forming single-stranded DNA. The next step is annealing, at which primers anneal to their specific complement sites at 55 to 65 Celsius, so that the Taq polymerase can initiate its function and build the new DNA strand at the final step, which is extension, and it's done at 72 Celsius. This was one PCR cycle in the thermocycler, which can be repeated many times. This was the general simplified principle of the PCR. If you have any questions, comment below and we will be happy to reply.